सही रहे ना अपने स्टूडियो ऑन ही है ना?
हाँ सर, स्टूडियो बंद सर। अभी मैंने उसी को भेजा ना।
उसी को मैंने लगा। ठीक है, let's look around.
सर ये स्टूडियो है सर, ये लिंक भेजा सर।
नहीं होगा कितने जना है? पार्टिसिपेंट्स स्कूल में
अभी सर अभी 4
ठीक इसको बंद कर दीजिए। Okay, so we are going to start today the concluding part of this systems biology class. Hold on for few minutes because there is some problem. Previously it was studio 3, then they moved me to studio number 1, so we have a little issue in streaming, so just hold on for some time. Okay, so yesterday we stopped it over here, that what is clustering of the data sets, right? I was trying to give you some examples. So today we have a real example where just a random example where I was showing you two different things. Like there's a person, there are 4 persons, in fact 5, yeah, there are actually. Yesterday let me switch this one off. Okay. So there are 5 person, A, B, C, D and E, just random person, and they had 2 primary expenses or primary expenditures which they do at the end of the day, spending on food or spending on clothes. Okay. Now these are the 2 items, and these are the arbitrary values, just for our ease of understanding the things. These are the arbitrary values which they actually spend in their food or in their clothes. Okay, and we are actually asking this question, that out of these 5 people, which 2 or which 3 or who actually spends or whose spending patterns are similar. So there are 2 people. Again, I'm telling you the question. The question is that there are 5 different individuals labeled as A to E, and they spend certain amount of money for food as well as for clothing, which has been represented at x1 and x2. And we are asking this question is that which spending patterns are similar, which people's spending patterns are similar, because there are 5 different individuals, so who of them are actually spending in a similar way. Okay, that's the question. Now the easiest way of doing that is doing what you call as a 2-dimensional plot. So just see the next slide. It's a 2-dimensional plot, and in this what we did is that we kind of plotted the two values which they spend on food and clothing, x1 and x2, and then how much they spend, the values of this one for these 5 different individuals, A, B, C, D and E. Say A spends, approximately 2 rupees on x1, which is suppose food, and then A spends about 4 rupees on clothing, and so forth. Now if I again go back to the previous one, okay, the previous slides, this is the one which I was talking about, like A spends 2 rupees on x1, 4 rupees on x2, B spends this much, C, D and E. And the question is that whose spending patterns are similar. Now once you do the plotting, you see very clearly immediately that the spending patterns for A and D are similar, and the spending patterns for C, E and B are similar, but they are different from the A and D group, right? And now we say that this is the first primitive example of doing clustering of the data. So clustering of the data is actually looking into the data and finding out the patterns within the data, which way the data behaves, what are the hidden information in the data, what are the things which are similar in the data, what are the things which are dissimilar in the data. Can we split the entire large data into smaller fractions, smaller subsets? If we can, what are the members of those subsets, and so forth. Why it is important? Because if you just have data and if you don't divide it into different partitions or different subsets or different groups or different fractions, then you are not going to get the full information out of the data, that what has been hidden into it. Okay, as and when we are going through this class, we are going to get more information on this. So very quickly from this particular example, we find that the spending patterns for D and A are similar as compared to the spending pattern of C, B and E, which are dissimilar. But individually this C, B and E are same, and A and D are actually same. Okay, so that is what I actually want. So this is the first example of doing the clustering of the data. Now, you could always say a very simple thing, and that is that look, if I had to do the clustering of the data, it's so easy. Why should I kind of go for systems biology class and do it? I mean, because if I look into this table, it is very clear that A and D are the same, similar kind of things, 2 rupees, 1 rupees, 4 rupees, 5 rupees, so they are kind of clustered together. And B, C and E are different, so they are actually clustered in a different way. So why do we have to even go for systems biology class to see something which is so simple and so obvious that I can just eyeball it, and then you can actually just find out that these are similar. Okay, that's the question. That's the kind of allegations which many of the people can say, that why do you need systems biology to do that? Well, the reason being is that this is an example of 5 different individuals spending onto 2 different items. Now transform this example into 5 different genes in 2 different conditions. Okay, if you are asking 2 different conditions, a normal cell and a cancer cell. Now tell me the expression changes of the EMT sets of genes. Suppose I give you a list of 5 EMT genes. EMT means the epithelial mesenchymal transition genes. Suppose I say E-cadherin, N-cadherin, vimentins, another 2 genes which are responsible for EMT, say TGF beta family genes, and ask you that, okay, these are the 5 genes, these are the 2 different conditions, tell me which gene expression patterns are similar. You could still do it, but think about in a real life scenario where we have 30,000 genes and 10 different conditions. Then you cannot do an eyeballing, and just looking into the data you cannot tell me that these are the genes whose expression patterns are same, and these are the genes whose expression patterns are dissimilar, right? So for a small data set, in other words, for a simplistic data set, so in a very simple world, you could still do it just by eyeballing it, looking it. But when the data set is like a gene expression pattern of 30,000 genes in 10 different conditions with 3 replicates each, then we are talking about something like, quite a big mega basis of data. And we cannot do that one just by looking into it and doing an eyeballing. Then you need a systems biology approach to break the data into what you call as clusters. So that is exactly what I had actually shown you in this particular graph, is that we did a very primitive clustering, and we see that just by applying the clustering rules, we could easily partition that into 2 different groups. So this is one group and this is another group. Okay, so that is the good thing about it. Okay, now I'm going to shift to the, is it clear everyone? Am I audible? Can anyone send me a little small feedback that I am audible, the slides are clear and everything is fine? Okay. Now we are kind of bringing into a term which we call as Euclidean space. What is that Euclidean space? Well, the thing it's just a big name, so just don't get kind of terrified with the name. Just bear with me. This is a very simple feature of whenever we make an XY plot in a 2-dimensional space. This we used to do it right from our high schools, right? So that you do it to two coordinates and then you kind of plot them. So this is known as the 2-dimensional space or Euclidean space. And in this 2-dimensional space, we can actually plot these values of A and B and C and D and E, and then I can easily do it. Now this is an example of 2 different points. So this is the example of 2 different points. This is one point, J points, and this is another point, I point. Just imagine, just why we are showing this one, why this small geometry, because we need to ultimately go back to our cluster programs. And over here, if I say that what is the distance between these two, then the distance between these two has been given by this Euclidean distance formula, which is xi minus xj's plus yi minus yj's. Or in other words, each one of these points will have an xy coordinates. So the y coordinate of this minus y coordinate of that square plus x coordinate of this minus x coordinate of that square, do an square root of that one. That is the distance between these two. See, remember, this is a formula which is not unique for this systems biology class. This is something which you had used in your mathematics in high school. So all we are trying to do is that we are trying to find out the distance between these 2 points, I and j, right? By using this particular simple formula, whatever I actually explained over here, right? Now if you understood that, now let's go back to our example. Let's go back to our example. And once we go back to our example, then each one of our example, we have 2 expenses. Think one of the expenses as x coordinate, another expenses as an y coordinate, and then we could actually always plot them in this way, which you had already shown in this one in the previous slides. Now all what we did is we are actually now use the same distance formula by calculating the distances between all of them. What is the distance between A and D? What is the distance between D and B? What is the distance between A and C, C and E, all the possible combinations of this one. And that is actually been represented in this case of this particular table where you do the distance formula calculations. Again, distance formula is using the information which we had actually in the previous slides. y2 minus y1 square plus x2 minus x1, and then you do the square root of that one. That's in high school mathematics, right? We use the same formula. Here the y and x coordinates for A are actually given, right? We know that what is the x1, what is x1 and what x2. So x1 and x2 is actually x and y coordinates. So we know that one, and then we are trying to find out the distance between these two. So this table, if you look into the table, the distance between A to A is 0, right? Because that's the same point. The distance between A to B is 6.325. A to B is 6.325. The distance between A to C is 7.0. So if you do all of this, calculating the distance, and from there you could see the distance between A and D. The distance between A and D is coming to be 1.41 for very less, right? So just by calculating the distance between the 2 points, I'm making a statement that just by calculating the distance between the 2 points in a 2-dimensional Euclidean space, by using the formula from the x and y coordinates of those points, we could find out the distance between them. And once we find the distance between them, we had already clustered it. without our knowledge, we are already clustered the data. Why? Because if the distances, now we could tell the algorithm that look, anything whose distance is less than 2 should be together. Anything whose distance is more than 2 should be thrown away in a different cluster. Now you know that the distance between A and D is 1.414, that's less than 2. So put A and D together in one bin, in one cluster, in one group, in one set. But anything which is beyond that, put them in the different sets, right? So see what is the distance between B and E. The distance between B and E is, suppose this particular distance, right? This is the B point over here, B and E, it is 1.118. So that's the distance between these two. What is the distance between D and C? See, the D and C, the distance is 8.246. That's a pretty large distance, right? So obviously by putting that filter that anything less than 2 should be all clustered together, more than 2 should be going to a different cluster, we quickly find out that these two can be clustered together and these three can be clustered together. So when you are doing a clustering program, the whole philosophy of this is that when you are doing a clustering program, the formula is written down over here. So when you are doing a clustering program, all we are doing is that we are calculating the distances between these 2 points. So even though people talk too much big thing about clusters and they may be sounding a little more terrifying to you, but understand that the core principle of them is the mathematics which you used in class 12. That is, we are just finding out the distance between these 2 points. If the things has a very little distance, then they are all together. If they are in more distance, they can be all thrown together in a separate group. But again, whether they are going to be together or they are going to be a different group is been decided by us. We are going to set the threshold. More than 2, one group. More than, if you can say that no, I want 5 as threshold. Then if it is more than 5, then even A, D and B will be together in one group. So what looks as to be in a different group will now be clustered in one group based upon what kind of filtering condition we impose into the algorithm. That is why the systems biology is important, is that you could make the data look any way you want it to look it, depending upon what kind of filters you put it, what kind of thresholds you put it, what kind of conditions you put it, right? Because I can always put in high conditions, and if I put, the distance should be about approximately 10 to be in the same clusters, then all A, B, C, D, E, F will be all clustered together as if they are all clustered in one particular group. So just by putting the different filters, different thresholds, we could make them to cluster together or separately, depending upon what is the biological question you are asking. Now, so far, I hope that we had understood that so far. I've been just talking about distance, and that might look very illogical to you, because what has distance to do in biology, right? Either way, if I'm trying to measure the gene expression signals, why should we bother about distance? Because there's nothing to do with my, what do you see distance in gene expression? Well, why the distance value comes is that each one of these points, when you do a microarray, each of these 2 points comes with their own fluorescence intensities. Remember, we put psi3 values, psi5 values. We had in red intensity, we have in green intensity. Yesterday when I had showed you that particular pixel containing graph, I should not go back because it's going to take a lot of time in this, because the computer is running at a lag. But just recollect that if you remember those slides, they are all this entire area of red color and the blue color and the green color, right? Red, green and yellow color. Now, if you think that red is an X and if you think that green is Y, then each of the genes will have a value for the red intensity and a value for the green intensity, right? Think that the value for the red intensity is X, and think the value for the value for the red intensity is X, and think the value for the green intensity is Y. Now that you have X and Y, you can put the distance formula. So just you can use the same distance formula now without doing the distance. All you are doing is that you are putting the intensity values, and these intensity values are a function of the expression of the gene, right? If the gene has been highly expressed, it will be possibly red in color. Why red in color? Because I explained to you, right? We took the cell, we are taking the cell genes, or we are taking the RNA from the cells which are grown in under 2 different conditions. Suppose one cell is growing in a normoxic conditions like this, another cell is growing in a hypoxic condition, say 1% oxygen. Now we are isolating the RNA from the hypoxic cell, we are isolating the RNA from the normoxic cells. Once you isolate the RNA, convert the RNA into cDNA, and the normoxic cell label it as red in color. Why? Because by adding a red colored fluorescence dye to those RNA coming from the normoxic conditions. Label with green color by adding a green colored fluorescence tag to all the RNA which has been derived from the hypoxic cell. So now we know that any green colored RNA is the RNA that extracted from the hypoxic cell. Any red colored RNA is the RNA isolated from the normoxic cells. Now I mix together, put them in a microarray, see what is the pattern of red and green color. Wherever you see more of red color, this means that particular gene has been actually overexpressed in a normoxic condition, because red is normoxia, right? And if you see a large amount of green in color, this means that particular gene, because there is more transcript, so it is the green color has overpowered it. So there is more color, more green color means more overpowering of that transcript. This means that is more in the case of hypoxic conditions. So if I talk about hypoxia inducible factor HIF, where you are going to see the intensity, whether you are going to see a red intensity or green intensity. Remember, the red color is corresponding to the normoxic condition, the green color is corresponding to hypoxic condition, and now we are growing the cells under hypoxia. So if you look into the hypoxia inducible factor HIF gene, then you'll see that HIF gene will be all green in color, because it has been induced only in the hypoxic condition. You won't find HIF genes into the normoxic condition, so the red signal will be less, the green signal will be more. Similarly, another hypoxia inducible factors, like for instance erythropoietin. We know that erythropoietin is also induced under hypoxic conditions, right? So again, you will see that erythropoietin gene is more of green in color, but there will be less in red in color, because erythropoietin gene possibly will be much less in a normoxic condition as compared to the other hypoxic condition. So with that colors, you have for each gene you can have the red intensity value and the green intensity value. For hypoxic conditions, the green intensity value is very high, the red intensity value is very low. But for a normoxic gene, say for instance, if you take any normoxic genes, we just take metabolic gene, suppose for instance, glucokinase. There is a metabolic genes which has been present in glucose metabolism, present in the normoxic condition. In that case, we will see that mode of glucose metabolism, this glucokinase has been present in the normoxic condition, so it will have a very high value of this red intensity and very low value of this green intensity. So each of these genes will have two different values, one corresponding to the green intensity, one corresponding to the red intensity. Plot them in the same plot and find out the distance between these two. Treat the intensities as distance, find out the distance between these two. So where the intensities are same, this means the expression patterns are the same. Because if the intensities are same, this means all of the gene expressions are similar in its nature, and therefore we can easily say that those gene expression patterns are similar. Now there is a term which you often use in biology that is known as co-expressed genes. What are co-expressed genes? Means the genes which are all expressed together as one operon kind of stuff. Okay, so most of the time those genes which has been regulating a particular pathway will be all expressed together. I'll give you an example. You take a cancer cell and treat it with cisplatin. Just you can do the experiment right in your lab. You take your cancer cell, in cancer cell line, if you are growing in the lab or your lab colleagues are growing it, take that, put it into a petri dish, add cisplatin into it, add doxorubicin into that. Now grow them for 2-3 days, right? You will see that genes which is related to apoptosis will be highly elevated, and they will be almost like 3, 4, 5, 10 folds, right? Because we have been adding those chemotherapeutic drugs which is going to induce apoptosis. So all those apoptotic genes are actually been induced. So if you see in a few of the apoptotic genes like BAX, BCL2, BAD, BIM, APA, cytochrome C, caspases, you will see that all of these genes will be highly elevated in those cells which has been treated with the chemotherapeutic drugs, doxorubicin or cisplatin, versus the cell which has not been treated with the drugs, right? So you see all the genes related to a particular pathway gets enriched, and these are known as co-expressed genes. Hardly we see one gene controlling everything except the case of master regulatory genes, which you see in the case of Huntington's disease, but majority of the time the gene works all in one network and all of the genes will be all together. So just by clustering you will know that which gene intensities are together, telling you that those genes possibly belongs to one pathway. If I do an hypoxia versus normoxia experiment, we'll see that all the hypoxic genes are been all clustered together because they are being induced in hypoxia, and all of these normoxic genes will be actually clustered together. So clustering is an efficient way of partitioning the data into different subgroups or different subsets, so that genes expressions belonging to a particular subsets are similar to each other and much different from the genes belonging to the other subsets. Again, I'm repeating this definition that clustering is a method or a mechanism or an analytical tool by which you create a heterogeneity within your data sets by dividing the data sets into multiple subgroups, subclusters, so that members which has been there in one cluster or one group or one subset are similar in their expression compared to the members in the different groups. because they belong to different clusters, right? Now once you do the clustering of the data, you basically breaking the entire data sets into numerous subgroups and looking into those subgroups and getting a whole lot of idea about the nature of the genes, whether they belong to the same pathway, whether they are co-expressed together, and so forth. So that's a fantastic leverage that you get just by doing the clustering of the data to know a lot about the gene functions. And that is exactly what is actually been done by using the microarray or next gen sequencing techniques and tools, just so that you're using this clustering programs. Okay, once you cluster it, I'm going to show you an example of actually clustering of the data and then doing a heat map out of it. You could see over here there are two different graphs. One of them actually show you the dendrogram kind of things, which is actually the clustering, where the genes belonging to, or having the same expression pattern are all clustered together, and the actual graph actually shows you the intensity in the form of a heat map, right? The heat map is actually, the scale of the heat map is actually showing in the bottom of the panel. I don't know whether, yeah, now it is visible in the bottom of the panel. You could see over here, when you say there is an overexpression, we are calling it by the red in color. Whenever there is a fold repression, down regulation, or decrease in the expression, then it is a green in color. Now you look and plot the whole thing with the help of a heat mapping programs like a heatmapper .org or something like that. You know, there are so many different tools to draw the heat maps. Once you make the heat maps, then immediately you will see that this particular gene expression pattern is divided into different color codes, and all the genes which are amplified together, all amplified, are all in red in color, and all the genes which is down regulated, repressed, expression goes down, are all in green in color, right? And then you could take all these green panels and then actually do what you call as in hierarchical clustering by doing a dendrogram, just finding out the intensities and distances between those intensities and plotting them on the two-dimensional Euclidean space, and then put them as one clusters by setting up a particular threshold, and all you get is this kind of things. Just by looking into this, you can actually say that okay, these are the gene expression patterns similar, and these are the genes which are actually been clustered together. So this is a wonderful way of knowing what is happening into the data sets, how the data set behaves like. Okay, so this is actually a real life scenario. This is taken from an actual experiment where people actually plotted the graph and then got this heat map, which is so much informative just by looking into it. Okay, now this is a few things to read. If you want to read about so far whatever we have discussed, these are a few suggestions. You can also find out so many different suggestions. Feel free to go and read your book of choice, but this is something which I feel pretty worth reading about understanding and getting a little bit of bird's eye view about what's going on, what you're talking, and that is actually been displayed on your screen. Okay, so this is what I was kind of yelling and shouting and trying to make you understand. Okay, this is something what is actually written in the definition is that the goal of clustering is to subdivide a set of items. In our case, the items are genes, right? In the case of items, in our case, the items are genes in such a manner that similar items fall into the same clusters, whereas dissimilar items fall into the different clusters. And this brings up two questions. First, how do we decide what is similar and what is not similar? And that is already we have explained that just by calculating the distance, if the distance is minimum, you set the threshold. Anything less than this much distance, this much intensity differences are going to be all together, and otherwise they are going to be put into the different clusters. One of the good examples of the clusters is actually when you go to walk to a shop. If you go to a departmental shop or in stores, in modern stores, then you'll see that all the items which is related to a particular applications are all clustered together. Say for instance, all related to the electronics, you have a separate electronic section over there. There, if you go and walk through, you are going to get A to Z everything of this electronics which is there. And if you leave that section, go to any other section, you are not going to find any electronic items. Okay, so this is not clustered together into one bin. You will see that soft drinks all clustered together. You will see that makeup and cosmetics, baby care products, and household appliances, electrical gadgets, all been clustered together. So all the people, what they're doing is actually on the background, what we did is actually we have clustered it. We have did and clustering in the, so that it becomes much easier when you walk to the stores. You don't have to kind of scramble into where this is, where that is. All it's been written is that clustered together into separate pockets, separate bins, separate aisles. You have to just go to that aisle, pick up your item of choice, and then come back. Right? It becomes much more easier. Life becomes much more simple once you cluster it, because you break the entire thing, entire items of the store into small tiny things, and therefore it is easy for you to access them. Okay, now this is something I think you have already understood the concept. This is the mathematics behind it, the clustering program. This is actually the way in which you find out the distance between these 2 points, or the distance of the intensities, or the difference in the intensities. Now over here, the formulae which has been written in the first 3 are the one which is in most commonly used. We call as the Manhattan distance, the Euclidean distance, and the Mahalanobis distance. So these are the 3 different ways of mathematical formulations which has been used to calculate the distance between these 2 points, or the intensity difference between these 2 points. Okay, this is the Manhattan distance, which is very commonly used. Euclidean distance, which I have already given examples by calculating the differences between the y-axis minus the x-axis square, and then square root of that one. And then this is Mahalanobis distance, after the name of the Indian scientist, statisticians Mahalanobis, and that is his way of calculating the distance. Now this is a little complicated because Mahalanobis distance has mostly been used in a non-linear systems in a 3-dimensional Riemannian space. We are not going to that, not to confuse you. There is a 2-dimensional Euclidean space, and there is a 3-dimensional Riemannian space. This kind of formula actually goes very good in those kind of in complex data sets. But for simple data sets in a 2-dimensional Cartesian coordinates in a Euclidean space, this formula is good enough to deal in most of the time. You don't have to remember too much about the rest of the things, but then the slide I will share with you, so feel free to go through the slides and have your interest, so go through these formulas, and if you are actually kind of want to know it deeper into it. So far the first 3 formulas are the one which is most commonly used, and for our case we are mostly using this formula number 2. That's something which is for the 2-dimensional Euclidean space, the distance or the difference in the intensities between these gene expression values between 2 sets of genes. From there we are going to do the clustering. I think we are actually clear with that. The next slide actually tell you that what are the actual real life algorithms which has been freely available. Many times you can write your own computer codes and do it. Many times you have to tweak the code which has been already existing. Many times you don't have to do anything, just put your data, upload it, and then the particular algorithm is going to do for it. So this is the one which are pretty much of them are all the open source cluster programs. Why I am listing it, because if any one of you interested in setting your hands on this one, feel free to visit any one of these sites, upload a data just like any random data, and then see how does the clustering works like. So cluster 3 program, ELK1, Octave, Orange, R, Origin script, and then SkitTims and Vikas and test clusters and Scavies. So this is the list is not ending over here. We have another 100 to 200 different programs for doing the clusters. Again, randomly don't pick up any programs just because you like it or like the name. Every program has its own applications depending upon what kind of things you wanted to achieve it. So that is very important. So we have 150 different programs, doesn't address the same thing. Each program is being tuned or geared or calibrated or equipped to work on a particular sets of the data, asking a particular questions. Just like you have the 3 different ways of calculating the distance, the Mahalanobis distance and then the Euclidean distance. So we had find out that this distance formula is mutually good depending upon what kind of data sets we have. If you have data in a 2-dimensional Euclidean space, Euclidean distance formula is much better, right? But if you see that the data is a little more complex in a different dimension, then we need to use a different formula. Just like that, the cluster programs, each has been tuned to work with a particular sets of data, asking a particular questions, right? You can't use any one of them randomly. So just that's the thing which you need to learn it. Now, for your purpose in this first curriculums of this systems biology, you can just keep on trying with your hands on, and then just see and play around with few of the programs and see how you are getting the feeling. We are not going to ask you in the exams any particular clustering programs and give you a sets of data and ask you to do clusters. You understand that that needs a little computational skill. For you, knowing itself is actually sufficient, because if you see majority of them like R and this in programming, and then this Orange is actually been written in Python. So Python is a sequence is a programming language. So many of them actually written into different C++. So you need to know those programs and then have a length and breadth of the applications of these clustering programs. Having said that, it doesn't mean that you cannot use it. There are many of these open source which are very free and very easy to use it. Use a few of them and then get a feeling of how the clustering works. So most of this clustering, what we do is actually hierarchical clustering, which are shown below, showing in the form of dendrograms, and partitioning. You are going to talk about partitioning now. So far you are actually talking about the hierarchical clustering. Now you are talking about partitioning. This is another way of breaking the entire data sets into smaller subsets and looking into them individually. That's one of the purpose of systems biology, is that taking into a large data set, starting with the large data sets, looking into the pattern which has been hidden into the large data sets, and from this pattern, whether you could predict what is going to happen, whether you have a trajectory which can be projected, what is going to happen in the next instance. Like if you have all the expression values of a particular gene, you could actually look them into different pattern and then say, okay, this sets of genes are actually elevated in hepatocellular carcinomas. These sets of genes are elevated in hepatocellular carcinomas treated with cisplatin, and which has a good response versus which has a bad response. So under every conditions, we have a particular set of genes which is either upregulated or downregulated. You can take each one of them and make a separate cluster, and each of the clusters can be used as a biomarker. Each of the clusters can be used as a diagnostic prediction tool. So a person actually works into a clinic, you do a gene expression profiling, you see this sets of the genes are elevated, and if you have an already clustered made on it, you say that okay, these sets of genes, if they are elevated, this means this person is going to respond very poorly to cisplatin. We are not going to give cisplatin, we are going to give this kind of drug, because either way, if you give the cisplatin, most probably the person is going to come back with a relapse after 6 months or a year, because we are already trained, we are already seen by mining lot of different data sets that the people who responds very poor in hepatocellular carcinoma using cisplatin as a therapy, they had this sets of genes which is elevated and this sets of genes which is downregulated. So just by following up what is your prior data sets and mining all those, and if you can make out separate clusters which has been aligned to a particular response, particular kind of a clinical response, a particular molecular response, you can have that stored into a computer, and the computer can actually take the decisions. All you need to do is that just feed the expression value into the computer. Computer is going to run them into the different clusters and see this patient's data falls into what clusters, and can immediately tell, okay, this patient is going to respond fantastically into platinum based or kind of or it's going to work into Vincalcolloids based chemotherapeutic drugs. They are not going to work on platinum based chemotherapeutic drugs, because these sets of genes are good responders for a particular kind of a treatment regime. So you can just have trained a machine, and that's the part of an artificial intelligence systems, where a machine can learn from your experience, and you can use this machine learning experience to do a prediction. You didn't have to be physically present over there to take the decision. Clinician don't have to sit over there to take the decision. The computer can take the decision just by training it with large number of data sets, a particular pattern. And how do you generate that pattern? By doing the clustering program, right? So you could see that there are so many different applications of this clustering. Now hierarchical clustering is already what you have seen earlier before, and then what you do is that we do a dendrogram out of that. You keep having tree-like structures, and then as I explained to you yesterday, possibly with an example, that suppose we take 3-4 medical colleges, and suppose we take AIMS and Safdarjung and Chipman Medical College, and then suppose RML Medical College, right? And then we do some kind of statistics and then find and see that okay, which are the medical colleges which has been associated with undergrad teaching. Okay, then AIMS is going to come, JIPMER is going to come, and Safdarjung is going to come. Okay, now whether are they belong to the same category, then we impose the next question is that which two medical colleges associated with undergrad trainings are proximal to each others by a distance of threshold, say 5 kilometers. Then even though in the first criteria AIMS, JIPMER and Safdarjung has qualified as a center for undergrad training, undergrad teaching, but once I pose the next condition, that is what is the distance which are proximal, put them together, then only AIMS and Safdarjung is going to be together, and JIPMER is going to be thrown out of the clusters, right? Because it has been very far away. Now if we add another conditions and say that okay, out of these three, which are not also associated with research activities, okay, then you can see that okay, AIMS has associated with the research activity, but Safdarjung does not have that strong research activity, whereas JIPMER has. Now AIMS and JIPMER will be clustered together because of the reason that both of them has the same kind of an agenda of a research activity. So you could actually impose different conditions and can pull out the sets which you are interested in seeing and which could be clustered together. So that is what is actually been important in these conditions, because these days when you see the data, these are very large data sets and you cannot manually look and find out a pattern within them unless and until we kind of impose the conditions and do this kind of a clustering program. Okay, now we are talking about partitioning. This is another way of breaking the data sets and looking into the different kind of patterns. Of course, there are so many different ways. We are just talking about these two, but then these two are not limited just by two. There are so many different ways, right? There is something called the Pca analysis, where we again take the entire data sets and then actually break them into different clusters, right? we know that what how to do the PC, even though we are not going to talk about this PC over here, but that is another way of breaking the data sets into different patterns. Okay. Now let's see in this case of partitioning of the data sets. So we had seen about the clusters. We are actually now we're working on the partitioning of this one. There are three different ways in which you could do the partitioning of the data. One is known as Eisen clustering, the second is known as k-means clustering, and third is known as SOM clustering. Remember, these are all, whatever I'm talking about, the clustering is part of this exam curriculum. So we kind of ask questions from there. So please read about them. Please make your concepts very clear about them, and then it will be easier for you to follow in the exams. If some kind of questions is given related to that, I may ask some questions that what is k-means clustering, tell me. Then you need to explain it with an example with the definitions and make it very crystal clear to the examiners. This is what you understand by the k-means clustering. What is Eisen's clustering and what is SOM's clustering, and so forth on them. Again, these are partitioning, right? This is a little different from clustering. Clustering is one approach of breaking the data sets into smaller fractions. Partitioning is another way of looking into the heterogeneity into the data sets. Okay, now let me pull into the next slide and let it to come over here. As I mentioned, these two are not the full one. There is something called third way, fourth way of looking into the data, and you can always divide them in. You can always use any kind of things to partition your data. So one of them, which I mentioned just now, the Pca methods, that is also a very efficient way of doing that one. That is known as the principal component analysis, where we have PC1 by PC2 axis, and PC1 by PC2 actually gives you an idea that what sets of genes are most important drivers of a particular gene expression pattern. Say for instance, so before the slide comes in, let me just talk a few seconds about this principal component analysis, where you have the entire data sets. And suppose we have a cancer cell which has been treated with chemotherapeutic drug and responded fantastically well. So this patient got this drug and she has been complete healed. There is no comeback of the cancers. Now this is another individual who has received the same drug, but then she came back with a relapse after 6 months. Now we are extracting the cancer tissues from both of these two individuals and looking into and asking this question is that what thing has happened in the gene expression profiling? What thing has driven that someone is a responder, someone is a not responder? Someone has responded so nicely that the cancer never came back, and someone responded so poorly that she came back with a relapse after 6 months. Okay. You can also always do in clustering. Now you can also see that suppose once you do, then you found that suppose there are 100 different genes which are differentially expressed in the responders versus non-responders. So in other words, these 100 sets of genes are differentially altered, are uniquely expressed in this responders individuals versus non-responders. Now you have this 100 list. Now you are asking this question is that, okay, these 100 genes are there with me in my hand, right? Now in this 100 genes, what are the ones which are most important drivers for this drug resistance? Because it's logical that all the 100 may be important, but we assume that out of this 100, a fraction of them may be the important drivers which drives the entire process of getting a resistant against a drug. Because many of the things are passenger, many of the things are drivers. Many of the genes are the ones which actually are important determinants for causing the resistance against chemotherapeutic drugs. Many other things follows with it. Those are passenger things. But the main drivers is the one which we should be interested in finding, because they are the one which actually drives this disease. So out of this 100 genes, which is differentially expressed between the responder population versus the non-responder populations, you do an PC1 by PC2 analysis and do in principal component analysis, you could see that those genes which are drivers will be all clustered together into one cluster, and those genes which are not the drivers will be clustered will be in a separate bin. Just by taking those drivers, you could actually say that, okay, these are the important determinants which has a profound influence, maximum influence into the gene expression pattern. So what you do is PCR by PC2 is that you find out that out of this sets of the genes, which are the genes particularly which has been responsible for this differential expression, which are the sets of the genes which is important for this differential expression and which drives this disease. So out of this clustered genes, Again, we are asking the next question is that which are the most important gene which has helped us to get this difference. That is what you do by the PC1 by PC2 component analysis. There is another way of looking into the data and breaking it into smaller components. Now this is an example of a couple of experiments which is actually shown over here. You could see that this is just a way in which we can actually partition the data into the different groups. Okay, look into the first one, this portion over here, the one where I'm trying to move my cursor. Okay, this one, this guy over here, this is a simple, we did a simple clustering, right? Now once we did a simple clustering, we could see that this gene expression, suppose this is in gene expression value, and we got it 4 different clusters. So in other words, in this entire gene expression of 20,000 genes, all the gene expression can be divided into 4 different classes. That's the way of looking it into in a simplistic way, that now we have the gene expression signature of 20,000 genes from a microarray experiments. What are the different clusters into them? There is just 4 clusters. So now from this entire 20,000, we have just brought it to just four clusters, or just 4 different groups, and all the genes in the particular group has a particular expression pattern. That's the reason they have a particular region in this 2-dimensional space, something over here, somewhere over here, and something over there. Okay, now we could say that okay, these 2 clusters are separate, but then the distance between these 2 clusters is also very minimal. So why don't we take these 2 clusters as one and make them one giant clusters? So what we are trying to do is that we are reducing. Now we are doing, now that you got multiple clusters, you are also seeing that what is the intercluster relationship between them, right? What is the intercluster relationship between them, and then trying to reduce the number of clusters to a very few handful. So from here now we can see that this particular cluster and this are very similar because their difference is very minimal. So we all put them into one large cluster, and these 2 are similar, so we put these 2 into one large cluster. Now from this 3 or 4 clusters, we have just 2 clusters, right? Now this is where I should stop with this one, because after this I'm going to go to the k-means clustering and come back to this one. But this is something what we are actually trying to do, is that we are doing an intercluster. So first we took the entire data sets and formed a clusters and got the clusters out of it, the different subgroups, the different heterogeneity, the different subsets, and then intercluster relationship we are actually been doing it by doing this partitioning program. So partitioning program is another step of refining of the data so that you can actually partition them into different groups. So we are talking about k-means clustering. So this is what is been actually explained in this k-means clustering. Let the slide to pop up in your window. Then I'm going to start with this one. Okay. Any doubts so far? Just feel free to email me, or just feel free to put it into the chat window. I can revisit the slide and explain to you again. Now, but that we are already heading at 1 o'clock, so just bear for me another 10 minutes. You should be done. We should be done with the systems biology class. 5 minutes I will take to kind of cluster all of these key points, and then we are good to go. Okay, this is the k-means clustering. Okay, what is over here? What we are doing in this one? Suppose these are the 3 different genes expression patterns. By clustering, we found that these are the 3 different gene expression patterns: a red pattern, a green pattern, and something like this, a red and green pattern mixed. Now what we are doing in this case is that we are putting a centroid. These are known as centroids. In k-means clustering, we are putting a centroid. Centroid is just a geometrical structure. Just put a centroid over here, and once we put a centroid over here like this, so assign each data points to the closest centroid. So randomly you put in centroids. You put one centroid over here, you put another centroid over here, and you put another center over here. Okay. Now what we are doing is that we are asking this question is that what is closest to the centroids? To this particular centroid, what is actually been closest? Okay. Now this data points are closest to the centroid, so we put them with this centroid, and these 3 data points are possibly close to this centroids, right? So now from this 4 or 5 data points, we have divided or partitioned them into 2 different centroids, right? One centroid containing these 3 points, because even though they are clustered together, they are not overlapping. There is a little variation in the values. Now we are going to look into that variations and putting all these 3 into one particular centroid, and all of these 2 into another centroid. Now once you have this one, then you have a large 2 centroids, one with 2 points, another with 3 points, right? Now we take this particular uh, clusters and then actually keep it aside. Don't do anything. Okay, now follow this one. Now this is another one where you have 1, 2, 3, 4, 5 genes expressions and put another random centroid over here, and then you see that these two are close to these centroids, but these three are not. So again we put them all together as into one clusters. And then you can also draw another centroid over here. So all what you're doing is that you're drawing an arbitrary random hypothetical centroid close to those clusters, and then finding out the distance between the individual genes with the centroids. If they are minimal with a particular threshold, you put them into one clusters, and you can keep on doing like this until you never find out any other gene sets which is away from one cluster or which is not been included into the clusters. This means you are actually you have completed analyzing all of your gene expression values, because now what you're doing is that you're partitioning those clusters into different groups, right? This is what is known as the K-means clustering. So K-means clustering, if you have to define it, it is the way of a centroid methods where you put a centroid, an arbitrary centroid close to those gene expression clusters, and try to find out that how they are getting partitioned into different groups, right? And once we did this, we repeat it. This is known as an iterative process. We repeat it and keep on repeating till we don't have any separate clusters. Now if you put these two with this x and this three with this x, still we have these two x together and then this one also. So now you are trying to include whether these two can be joined together like this. Okay, as I given you an example yesterday, that AIMS of the Jung RML JIPMER, AIMS and JIPMER, AIMS and Safdarjung can be related by the undergrad teachings. AIMS and JIPMER can be related by an applications of research activities. Okay, fair enough. But we found, suppose the cardiovascular department of Safdarjung is very good, similar to that of the cardiovascular departments of this RML, but not so good. But AIMS cardiovascular division is not to that extent good, and the JIPMER is also not that good. So now we see that Safdarjung has been now clustered with the RML in terms of the cardiovascular department or the cardiology department over there, right? So now they are actually been related to one clusters. Now we say that okay, but we already linked AIMS with Safdarjung because of the proximity in the distance and because of the reason that both of us actually are engaged in undergrad training. Now the Safdarjung is also related to RML because both of these cardiovascular department or these cardiology departments are very good. So in a way AIMS is also connected to RML. So why don't we put AIMS, RML and Safdarjung into one clusters? And we know that AIMS is already clustered with JIPMER in the terms of the research activities. So now that AIMS and RML and Safdarjung are together, we can also put JIPMER into that clusters and form a very giant clusters. So all what we are doing is that we are trying to see the relationship between between variables, right? And we keep on doing it till we find that there is no member is actually be left over. That actually gives you a particular relationship between these two, between all the data sets. Now over here you might have a question that with the help of clustering, what you are trying to do is that you are going to break the data sets into smaller fractions. Now we are talking about joining the data sets. Isn't it that they are mutually exclusive? No. So initially what we do is that we find out the pattern within these data sets, right? So that is the whole purpose of doing a cluster. So again, I'm repeating the questions. It might come into your mind that when you talk about clusters, we are talking about breaking the data points into functional units and then actually trying to find out that what is the pattern into the data. But when you're doing this one, then we are trying to again join together and then actually find out the relationship between this. So isn't that they are actually in an opposite directions? No, because when you do the clusters, we try to find out the patterns within the data sets, number one. And then in order to find out the relationship between those clusters, intercluster relationships, you need to use this kind of programs, which is known as the partitioning, so that you know what is the relationship between these different clusters, how one cluster is being related to the other clusters, right? That is the whole purpose of doing the partitioning. One way of doing that partitioning is what is known as the K-means partitioning or K-means clusters. Okay, this is the hierarchical clustering, which we already discussed about. I'm moving away from this one. We are almost done with this one, and this is an example of, this is a real life example. This is an example of from an insurance company, a company which actually provides insurance. They actually has given kind of this example. I'm just telling you, it's a little funny, but I mean it's very easy to understand. There is two different insurance companies that is known as Allstate and Geico in United States, and they had studied the pattern of accidents which has been happening on the US roads. So we are giving an example a little away from our biology, so that the concepts actually becomes very clear, and also to make you understand that these are very universal tools and techniques, need not necessarily been used by the biologists only. You can actually use it for any kind of other things, in a social experiment, sociology, epidemiology, some kind of a things. Wherever you need these kind of questions, you can actually use the same tools. That's the whole purpose of actually telling you this. Now, suppose they found that the incidence of road accidents with the male versus females in New York. That's the questions, what the query, that's the hypothesis, that's the question which they are actually asking. They found that males are 40% more likely to be involved in an accident. Males are 40% more likely to be involved in an accident than females. So we know for sure that males are actually more prone for doing an accident when they drive a car than females. Okay. Now they find out that, okay, what is the pattern of those people who has actually caused accident, who has been engaged in an accident. Then they found that the males with accidents are scored for age and race. That's the next thing. So okay, these are the individuals, these are the males who has committed most number of accidents on the New York road in the last year. Okay, what is the pattern of them? Okay, let's cluster them into age and race. Then they found that males within these particular age groups, 25 to 30 years, are 65% more prone for accidents. Or in other words, the youngsters are more prone, are causing more accidents into the road. So if you see the demography and if you see the age and sex and gender, you could know all from the information from the police records, right? And they found that males within this age group of 25 to 30 years are 65% more prone for causing accidents, like than males. We're not even talking about females, than males of the different age group. Okay, fair enough. Now let's see on the basis of the race. Then it is found that males who are not white or black or African Americans, those males are 17% more prone for accidents than white males. Or in other words, in most of these accidents we found that it is the black persons driving the car or the African American who is not white are the one who have been engaged in more number of accidents. Okay, what is the next informations? That among all the things, so far you had partitioned it according to their sex, male or female, according to their age groups, 25 to 30 years, according to their race, which is white or non-white, and then also according to the car color. It has been found that males driving bright colored cars, bright red, dazzling red, very weird colors, which normally most of the people don't drive. Males driving bright colored cars are 70% more likely to involve in an accident than those driving dull colored cars. Because this goes into psychology. So this is not just an example. When you train your computers to look into this kind of data for the last 10 years, you emerge what emerges out of it. What emerges out of it is a psychological predispositions. What is the psychological predisposition? That black males within this 25 to 30 years of age driving a bright red colored cars are being more prone for accident than females, than white males, than people who are not driving colored cars. So any male, so once you have conditioned our systems with these preconditions, now any new entries, any new person who is purchasing, trying to come to my company to purchase an insurance, I'm going to check these things: age group, I'm going to see whether it's in black or white or non-white. I'm going to check whether they are driving a bright colored cars, and I'm going to see their genders. If I have these 4 different determinants, 4 different matrices, then I'm going to charge them more in the insurance, because I know that those individuals have been engaged into more number of accidents in the last 10 years as compared to the individuals who are not in this. So black males in the age of 25 to 30 years driving bright colored cars are charged with a monthly EMI, because when you do an insurance you have to pay an EMI. Monthly EMI is 15 to 20% more than the rest in the group, right? And this, why so if you are filling an applications and you find that you have suddenly charged 20% more, you can go back to the company and ask, why am I being charged more? The reason being is that there's a 10 years of research behind it, where you found the patterns of the accidents and you found that you fit to this pattern. So therefore you have clustered together with the help of this clustered together with these individuals whose way of thinking is similar to yours. So this is kind of a psychology. That's what I'm saying. This is a kind of a psychological predispositions, and it has been really proved by scientific fact that if you write like bright colors, dazzling reds, and if you have been at particular age groups, and if you have a particular likings, you are being driven by particular psychological features, right? And that actually matches to the people who actually likes to similar kind of things. So you can all put them into one bins. I'm going to give you the next example, and it's going to be even more clear to you. This is an Amazon trip for binding, for purchasing different books. Now, how many of you has actually encountered this scenario that You purchase one book from Amazon, suppose an e-book or a real hard copy book by ordering it from Amazon, and after that you found that the moment you place the order, or the moment you are going to place the order and going to put them into the cart, then suddenly you get a notification from Amazon and tells that customers who purchase these items in a shopping cart also brought this. Like most of the time I get this one. I think every one of you do it, because the Amazon has actually been running a clustering program in the background. So they found that people who likes this particular things, like the molecular biology of the same by John Wilson, those people also purchased, a few of them also purchased either one of these three books. And therefore, if you purchase, suppose this book, your psychology or your liking is possibly belongs to this category. So why don't we recommend these additional books to you? And it has been found that most of the time you end up in purchasing one of these additional books also, even though you initially didn't logged into the Amazon site with the aim or with the intention of purchasing three books or two books, just one book. But then you found that, oh my god, I suddenly like this book also. And why they had given out of this millions of the books in the inventory, why they had given these three books? Because people who purchase these books also most of the time found to be purchasing one of these three books. So you belong to this category and they give you that options, and if you fit into the option, there is another data point into their survey that okay, we are true. So this is a part of this artificial intelligence, part of the machine learning program, part of this robotics, where you train an equipment and an instrument to take a decision on what is the psychology, what are your thoughts. But core of this particular decision making thing is clustering. All you are trying to do is that you are trying to put the peoples of the same psychological thinking in binning them into one clusters. The same thing happens in the case of gene expression profiling. You're trying to profile all the genes belonging to a particular pathways, say differentiation pathways, EMT pathways, apoptosis pathway, proliferation pathway, drug resistance pathway, all putting them into different clusters because all the genes behaves in a similar manner. Just like the people purchasing this book is going to behave in a similar fashion by ending up purchasing similar books, that's the same thing about clustering. So clustering is all about grouping the data, finding out the pattern into the data and putting them into isolated bins so that you could very well partition the data into different groups. Okay, so that actually explains the clustering. I'm not clear. If I'm not clear, you're going to come back and ask questions. Feel free to do that one. So how does the find the relative things? Customers. I mean, this is just the algorithm which is actually showing over here. I better not explain too much because already we talked a lot about this one. So I'm just going to quickly skip this slide. The moment it pops up in your window, I'm going to remove this one. This is just the way in which the algorithm has actually worked. So let's remove this one. Okay, this is the one which I was trying to show you. It's possibly in your screen. So again, this is the work, this is the mathematics or the statistics which has been going behind it. The same clustering things. Okay, which one is going to recommend and which one you are not going to recommend. Related, the same thing, you just now replace those Amazon stuff, replace those car insurance stuff, put your genes. You are seeing that I was talking about co-expressed genes, remember 10 minutes, 15 minutes back? That's what you're talking about, similarly expressed genes. You use a different language, but the meaning is exactly the same, co-expressed genes. So all of the genes which are co-expressed under a particular condition. Say for instance, you take a cell, grow it in hypoxic conditions, or take a cell, and add it to a particular cytokines. What are the genes which have been expressed or whose expression pattern follows in a similar trend? We call them as co-expressed genes. And majority of the time it is found that all of these co-expressed genes belongs to a particular signaling pathways, forming what you call as a signaling operon or signaling cascades. Because in the cell at a given point of time there will be hundreds and thousands of signaling going on, but all of the signaling cascades are all uniquely separated and segregated from each other, so that there is no crosstalk between these two. Now you found that all the genes which are similarly expressed possibly belong to one clusters, possibly belongs to, why you are saying possibly? Because you have been after all predicting it, right? You're not actually doing an experiment, you're just predicting it from an experimental data. So possibly these genes all co-expressed together belongs to one particular pathway, and most of the time it is true that if it is an hypoxic condition, you will see that majority of these genes which has been co-expressed belongs to this hypoxic pathway. Almost 9 out of 10 genes will belong to the hypoxic pathway. And one of the way of doing that one is something called the David pathways, the Goa pathways, the Ingenuity pathways. These are the different pathways where you feed the gene expression values and the name of the genes, and they're going to tell you quickly whether they belong to a particular defined pathways or not. So there is the David pathways, DAVID, there's the Go pathways, there's an Ingenuity pathways. There's so many different ways to do the pathway analysis and finding out what are the genes which are co-expressed, and what kind of biological pathways do they converge to. Very important for you to know you. Okay, now we are not talking too much about this, and I think so we had reached to the end of this class, which is a little elongated class, I mean extended class, and kind of bugged you for 2-3 days. But this is something very important. We are going to give you an example, last example, and before that we are going to kind of separate it. Just bear for me another 5 minutes. This is something called the false discovery rates. This is why I have brought, because of the reason that there are two types of errors. I assume that most of you are aware of this type 1 errors and type 2 errors. Just go and read. We don't have too much time to talk about this in detail, but we have this false positive errors and this false negative errors are there, this type 1 errors and type 2 errors. Now what we are trying to say in this particular example is that most of the time we kind of judge the value of our data by putting in p-value, right? The p-value should be less than 0.05 to be statistical significant. Anything less than p05, we actually assume it to be universal truth, and we are actually assuming that that's statistical significant. Well, there's a little bit of caveat for that. This slide is just to tell you and flag you that they don't take the p-value for granted. P-value is 0.05. This means that if you perform an experiment 100 times, 95 times you are going to get the same results, and 5 times there will be a deviation, right? Now this p-value 0.05 is pretty good for a small low-tech experiments where the number of samples is very less. But think about it, where we have 20,000 gene expression data, because now we are talking about systems biology, so we have one expression of 20,000 different genes. Or you did a mass spectrometry, we have a profile of about 100 or 200 or 5,000 different proteins in the proteome, right? Now we cannot talk about p.05, because even if you have 5% error, 5% error for about 10,000 different genes is going to be a large number. Those many number of genes will be false positives for your case. So p05, all I'm trying to tell you is that p.05 is a very good for small data sets. But if the data set is very large, where there are a number of entries, 20,000, 30,000 different genes, different proteins, just if you use p.05 is not going to work, because even if we have 5%, 95% will be true, but 5% of that 30,000 is going to be a large number, and those many will be your false positives or false negatives into the data sets. So you cannot use just p is equal to 0.05 and do just simple p-value calculations. You need to do what you call as a false discovery rate, or FDR. FDR is another way of making the p-value very tight, so that out of this 0.05 we further filter it and actually reduce the number of entries and make the data sets more stringent. Again, this needs possibly discussions. We are free to do it whenever we meet next time, or put your questions. Now very quickly, 2 minutes, we are going to wrap up the class with a summary. So systems biology is an approach of analyzing the data. It's a mindset, it's a psychological way of taking the data and looking into the data and finding out that what is the maximum information is hidden into the data. Unlike conventional biology, systems biology starts with the end of the experiments in a way. I mean, of course there are experiments beyond that. So if you do in conventional experiments, you do grow the cells, get a large data, and then you are done with that one. In systems biology, you start with the large data, and then you try to find out a pattern within this data, whether this data belongs to a particular, whether it's going to show in a particular trend, whether there are different entries into this data which can be visualized together, and all these sorts of things. So systems biology is more of an approach rather than actually doing the experiments, right? That's a kind of a mindset. Now in the first class also we talked about the different ways of generating the data. Now we had also seen in the last class, okay, once you're doing the systems biology, about a little bit on microarray and what is microarray tools and techniques. Why? Because these are the ways in which you generate the large data. And today what we did is that we analyzed that what is the pattern hidden in the data by doing what you call as in clustering, and within that clustering to find out what is the relationship between the clusters. For that what we did is the partitioning. So partitioning is a way by which you find out the relationship between the clusters, and clustering is the way in which we try to take the entire data sets and divide them into smaller subsets, so that we know that what are the entries, what are the different genes which belongs to a particular pathways. This is evolved from a concept that is known as co-expressed gene, because if you do the clustering, you get a fairly good idea that these sets of the genes are actually been driving a particular biological pathways, and all working together just like an operon. So that is the most important aspect of it. Again, having said that, this is just a prelim, very basic introductions for systems biology. Nevertheless, it is not complete. We need at least another 1 month of the classes, tools and techniques to know the full length and breadth of the systems biology. But having said that, this is a subject that heavily needs computational biology, mathematics, physics, and biological experiments. So this is a little bit more in a non-conventional biology which you are used to do it, but at the same time it is extremely important, because this actually gives you an idea that how much data is actually been sitting into the public domain, and you could use this data and analyze it in a systems biology approach and get meaningful solutions out of it. That's the whole purpose. You don't have to do every single experiment yourself. You can go to a public repository, take the data out of that and do a fancy analysis of this data and look it in a different approach by using the systems biology tools and techniques. So with this, very sorry to hold you for the next half an hour beyond the class, because I am not going to take any further class. So we are done for today, and that actually ends our systems biology block. I'm going to pass on to you all the slides, and if you have any queries, any doubts, just feel free to email me, WhatsApp me, and SMS me, whatever the way you could reach to me. I'm very reachable, so I'm just in the campus or in the department most of the time, you know. So just, you know, we can always clarify these doubts and, you know, kind of progress it further. From the exam point of view, you need to never forget the definitions of the systems biology, which I actually studied in the first class. You know, interrelated, interconnected, interdependent, interacting systems. We talked about boundary, interface, chaos of the systems, linear systems, non-linear systems, predictability. Those are the basic fundamental 101 of the systems biology, so don't forget that. That is very basic, and these are a little more advanced in that directions. Okay, so with that we end the class. Thanks, stay safe, and again, I'm absolutely thankful to you because you could spend some extra time to be the part of this class. So thank you, and pose your questions. If you have any questions, just feel free to write to me.